Hello friends, this is Dr. Chaitali and welcome to my YouTube channel Physiology. Today we will perform a very simple practical which can be useful during your practical examination, that is determination of blood group. Here we will only deal with ABO and Rh system and will perform only practical. We will not cover the theory portion, so let's start. For this practical, we require antisera, spirit, cotton, lancet and glass slide. These are antiseras. Blue one is for anti-A, yellow one is anti-B and transparent is anti-D. First of all, clean the finger with spirit swab. Then after, prick your finger with lancet very carefully. Here we require only 2 to 3 drops of the blood. If blood is not coming out, so you need to do milking process. By this you can get enough amount of the blood. Here we have taken 2 samples on each slide for easy understanding. You can do with only 1 sample on each slide. Now name the slide as A, B and D. After that, add antisera A in slide A. Antisera B in slide B. And antisera D in slide D. Now mix antisera with your blood or a sample properly and look for the agglutination. Here in slide A we can see that blood mixed with the antisera and there is no clumping or no agglutination. Let's check for a slide B. Here in a slide B also we see that blood mixed with the antisera and there is no clumping or no agglutination. Let's check for the slide D. In slide D, we can find clumping or agglutination of RBC that can be seen as a dot-like, very fine dot-like structure in the slide. If you are getting confused with the agglutination, you can compare with its negative slide. So our result is a O positive blood group, because there was no agglutination in a slide A and B, while agglutination was only present in a slide D. Let's check for the second sample. Here we can see that there is no agglutination is in slide A. While there is a agglutination in a slide B, we can see the clumping of RBCs. Slide D also shows the clumping and agglutination of RBCs. So, in the second sample, slide B and slide D shows clumping of RBC. The blood group is B positive. Now we are repeating the same procedure for the another sample. Let's check the result. So there is a agglutination in a slide A. We can clearly see the agglutinated RBCs. There is no agglutination in a slide B. And there is agglutination in a slide D. So the blood group is A positive. Let's examine one more sample. In this sample, we can see agglutination in slide A as well as agglutination in slide B and the agglutination in slide D. So the blood group is AB positive. Let's check one more sample. In this sample, we can see that there is agglutination in slide A. But there is no agglutination in slide B. As well as no agglutination in slide T. So we can conclude that blood group is A negative. Let's quickly revise it. In sample 1, there is no agglutination in slide A and B and only in D. So the blood group is O positive. In sample 2, there is agglutination in slide B and D. So the blood group is B positive. In the third sample, there is agglutination in slide A and slide D but not in B. So the blood group is A positive. In a fourth sample, agglutination in all the slide A, B and D. So the blood group is AB positive. In the last sample, agglutination is present in only A slide. So there is a negative blood group. I hope that all of you find this video very helpful and able to perform your practical. Stay tuned.